This is my poster board for the protein synthesis. In the process that goes from DNA, which goes through the transcription, links with the the mRNA, goes through translation, and finally form a protein that either stays in the cell or leaves the cell for exocytosis. The process begins with transcription. And here I put that the initiation is the first step of transcription, which begins with the promoter. It binds to the transcription factors and the RNA polymers, which is here. And the elongation is what I tried to depict here. I kind of copied off of the book for reference, but once these separate, the mRNA is what tails off. And then there's the termination, which happens whenever we get to the end of the gene, wherever that is. Which here, I've depicted it here. Then you go through transcription, which again, I kind of copied this out of the book, but this is the amino acid along with the cell coding, and at the bottom the anticodon is formed, which links with the codon in the process, which is what I was going to talk about here. I kind of drew it out to explain and also to help me say what the process is. The DNA is formed in the nucleus, which I did a depiction here, so it's kind of hard to see, but this was my step one, and my pen kind of smeared, but it all starts with the DNA, with the nucleus, which contains the DNA, which is copied to make all of it's the skeleton key that produces everything to make other proteins. So anytime a protein needs to be created, it comes from the DNA cell and it replicates the DNA to form the new protein needed. So then once the DNA is found in the nucleus, it is copied by the mRNA. Okay, so once the mRNA is created, then it's transferred to the ribosome, which I've drew my number 2. This is my ribosome. And there, the tRNA also comes, and then they link the codons to the original recipe, which was the copy from the mRNA, and they form the polypeptide chain, which is my number 3. That's my polypeptide chain. And then the polypeptide chain is the protein. So this is where it becomes to the translation. And if modification is needed, I've went further. It then goes to the ribosome will transfer to the rough ER, and then the protein will be folded, and once it's folded, it'll be sent into a vesicle that will then send it down to its final destination, which is the Golgi apparatus, and there the proteins are prepared to travel outside of the cell for whatever they're needed for, or if it's used to replace a cell inside the cell that's been damaged, it'll go there. And that's the whole process.